So yeah, hello everyone, and thank you for joining us for this webinar, where we basically want to introduce you to the new Visium HD technology. So for those of you familiar with the Visium assay, this platform enables you to perform whole transcript analysis with a spatial context. So basically, we are looking at gene expression in its natural location within a tissue. Visium HD does exactly that, but with a much improved spatial resolution. So this new technology allows you to explore gene expression at a single cell scale, basically. The new slide design does contain capture areas with a continuous lawn of 2 by 2 micrometer barcoded squares, and that allows for this improved resolution. In addition to the improved slide architecture, this assay is also enabled by our Visium CytAssist instrument to ensure the accurate localization of the transcripts that you hope to capture. Taken together, what that new technology can do can be seen on this slide in a pretty striking way, I would say. On the left, you see a colorectal cancer sample done on the first version of our Visium assay containing the oligo-covered spots. So if you remember, these spots have a diameter of 55 microns and are 100 microns apart center to center. And you can see from the clustering shown here that different tissue types can clearly be discerned by their transcriptome profile. So what you're looking at here is not just a morphology image, but the clustering based on the transcripts. So this already shows the power of spatial transcriptomics, but the same sample type was also examined with Visium HD, and the results of that can be seen on the right here. The resolution is clearly advanced, and the results are much more detailed. And you can see just how much the clustering based on transcriptome reflects this high resolution. So based purely on the clustering, you can pretty much see the tissue morphology in a great detail. So if you are interested in looking at these data sets, you can find them online. I will show you later where to find them. This is just kind of to tease you and show you, if you compare this, how much the resolution has improved. So how was this accomplished, basically? So practically speaking, in the Visium HD workflow, the first 3 steps, so the sectioning, the staining, and the imaging, take place on the standard histology slides, followed by a transcriptome probe hybridization step, which is an overnight step, after which these probes that have hybridized are ligated together. And then in the next step, the 2 glass slides that you started out with and the Visium HD slide are placed in the CytAssist instrument, which aligns your tissue sections to the Visium HD capture areas. Then an image will be taken in the instrument to show the orientation of your tissue relative to the capture area. So you can analyze your data later on. The ligated probe products are then hybridized to the Visium slide. And once this step is complete, we go off instrument, start with the probe extension to extend the oligos we need, which are then carried through the library construction to prepare a sequencing ready library, which can be analyzed then using Space Ranger and Visualize Loop Browser or any third-party software that you want to use. So I mentioned the slide already earlier, and now I want to show you what these exactly look like. You can see here on the left that each Visium HD slide contains 2 capture areas, and each of these look as shown here in the second image. The lawn of oligos, so this lawn that I mentioned before, is 6.5 by 6.5 micron square that is surrounded by this fiducial frame. So if you are familiar with the first version of Visium, you know what a fiducial frame is, but you also know that we'll see that it looks much different compared to this. But this frame is necessary to be able to locate your transcripts on the tissue itself. So as I mentioned earlier, an image will be taken in the CytAssist. And when we look closer into the oligo lawn, basically, this is what I'm speaking of now, we can see that they are arranged like this grid that we can see here. It's a continuous grid of barcoded squares with a size of 2 by 2 microns. And by default, these are binned to an 8 by 8 configuration because that usually is a great starting point for the analysis itself. And the oligos themselves, so I made this a little bit smaller so we can fit this information here. The oligos themselves contain a TrueSeq read1 sequence, the UMI, so the unique molecular identifier, a spatial barcode that is shared within a 2 by 2 micron square. And together, the UMI and the spatial barcode are 43 bases long. And finally, we have a poly-T tail that enables the capture of the ligated probes. And remember that we are transferring the ligation products from the glass slide to the Visium HD slide. And this allows these products to be captured where they are located. The probes that are the basis of these products that we need to capture have been designed like you can see here. So they have a left-hand side, they have a right-hand side. And the left-hand side contains a partial read to S and a sequence that binds the target RNA. So it's just depicted here in this purple color. Likewise, the right-hand side contains a target sequence as well, and a poly-A tail that will then be captured by the poly-T tail of the oligos that I just mentioned. So this is basically what happens during probe hybridization. They find their target site and sit down where they belong. So we are working with, for HD, with the version 2 probe sets. They are for mouse and human tissue, and they are whole transcriptome probe sets. So they contain 18,000 genes in the human set and 19,000 genes in the mouse set. And these probe sets are designed in a way that most genes are covered by 3 probe pairs. An exception there are genes that are generally very highly expressed. So these have only 1 probe set. On the graphic on the right, you can see a representation of how the 3 probe pairs are designed and where they can be located on this gene here as an example, but they are spanning the genes, so to enable the most accurate hybridization steps. So I said it earlier in the beginning, to perform the Visium HD assay, we do need the site-assist instrument, and we need the instrument to be updated to the firmware. It says 2.0.0 or higher. So this is what you need to do to be able to run the HD workflow. The workflow itself, or the runtime, is still 30 minutes. To basically transfer the probes from 2 tissue slides onto the 1 Visium slide. And the instrument needs to be connected to the internet to be able to download this necessary firmware. This can be either Ethernet connection or Wi-Fi, both work completely fine. And honestly, this makes the upgrading extremely easy to handle. So when we look at the assay itself, it can be broken down to several parts. We usually start out with the standard histology slides. We perform standard sectioning and placement of 5 micron thick sections. So this is still the recommended thickness here on these normal slides. And the sections are then stained and imaged in an appropriate way. So based on your staining, bright field or fluorescent imaging. So basically at this stage, all we need is, and all we do is standard histology. When we move on beyond the staining and imaging, we go to the molecular part of the workflow, basically. The sections undergo an overnight hybridization of the probes that I showed you earlier, that are subsequently ligated after this overnight hybridization is finished. And only after ligation, we move on to the barcoding slide. So that's basically a representation you can see here in the middle. And I think the image on the top right here shows pretty nicely how the orientation of your 2 original slides shows up on the actual Visium slide. So this is the configuration how it would be in the instrument itself. You have two tissue slides, sample slides on the top, and you have one slide that the probe products get transferred to. So the concept of this cytosys is pretty easy when you can picture it like this. You have two original slides, and the products of these two will then be transferred to the Visium slide in the machine. So I hope this makes sense. I just liked showing this image because it shows the concept pretty nicely. So why is the cytosys such an essential part of this essay and basically good Visium data? With our first generation Visium approach, the spatial precision is limited by various factors. So for one, the reactions are taking place in a liquid, which by nature allows targets to be released above the tissue as well as below, which can lead to loss of molecules. In addition, the targets can also be released between features. So basically what you can see here and be lost that way because there is no capture possible in between these oligo-covered spots. So with the cytosys instrument though, a lot of this uncontrolled loss and loss of capture is eliminated. The reactions take place in a tiny space between the slides containing the tissue and the Visium slide. So thereby it's minimizing the risk of losing transcript because they swam somewhere else. In addition, the Visium HD slide capture area does not contain any spaces. So as I mentioned before, you have on the older version, you have these spaces that are not oligo covered in between. This does not exist on the HD slide. So the products can be captured wherever they are located, and they don't kind of need to be lucky to be located where oligos are, because for the Visium HD, the oligos are covering the entire capture area. So the cytosys instrument is critical to prevent the loss of molecules and to get the best data out of your precious samples. And of course, if you have a continuous lawn of barcoded spots or barcoded squares, we do not want to lose anything or let anything go off the wayside here. So this is why the cytosys is critical for this. Let me now say a few words about the sample preparation for the Visium HD assay. So right now, Visium HD is out for FFPE tissue. We started out here because this is the most used and archived sample type. So as you can see, we have tested a number of tissues already. But if you have a sample type in mind that you cannot find on our website, we do have a wonderful support team. So if you have a specific question for us, we are more than happy to help you. We have tested not only FFPE blocks that we sectioned fresh, but we also tested archived slides at different storage conditions. And we see that all of these samples showed very similar results when we look at the UMI distribution. So what you can see here on the left is a result from a freshly mounted tissue section. Here it was archived at room temperature, once at 4 degrees, and once we archived it in a hard cover slipped way at room temperature. So this actually, this storage condition happens a lot in the field. So this is why we wanted to take a look at this here. And we see that we have a high correlation of our results between all these storage conditions. So, but of course you don't need to start with an FFPE block, but you can start with an FFPE block if you have them available. So once you start out with a block, the first recommended step to take is to perform tissue QC. So what that means is basically you can cut a section onto a normal slide and see, okay, does this block even look nice enough for what I want? And then what we recommend is to run a DV200 analysis. And we see that samples with a DV200 above 30% have very robust and good results. If it's lower than this 30%, it's a bit risky to use. So if you have different blocks available, maybe at this stage you should consider switching. If not, please be aware that a DV200 below 30% is a little bit of a risk. If it's above 30, then yeah, just go ahead, go ahead and use your block. If you have archive sections on slides already that are stained, so then in that case, you would have to take a look at where on my slide is the section that I'm interested in. And would this work with aligning it to the Visium slide and to the capture area? If it's in the allowable area, I will show you later how you can find this. We do recommend to look at the DV200 as well. So this is, of course, a little bit tricky. But what you can do is you image your archived slides to see if the morphology is still up to what you want. You then remove a hard set cover slip. This can be done really carefully, and we have some protocols for this if you're interested in knowing how this works. Scrape some tissue, either from similarly stored non-experimental tissue. So, you know, you have 6 slides available, 4 of them do not work with the alignment. So these 4 would be perfect examples or perfect samples for RNA quality assessment. So calculate your DV200 of the RNA of your curls that you can take off them, and then you would proceed with the destaining. So this is just to show you that if you have a lot of samples that are archived and hard cover slipped, you can definitely use them. So when you go on from cutting your own sections and going on to staining them, of course, you can do H&E staining because this is the most common stain for tissue morphology and in pathology. So we use hematoxylin, bluing buffer, and eosine. We have tested the GIL2 in hematoxylin, and it is actually recommended because it gives you really nice images here. We do use a bluing buffer to darken the hematoxylin stain and the eosine to stain our basic structures as well. Of course, what you can also do is do immunofluorescence staining if H&E staining is not suitable to your experimental needs. So then you can detect the localization of certain proteins you are interested in. So this immunofluorescence staining can be either direct. So the direct version detects the proteins of interest when the fluorophore is conjugated directly to the primary antibody you are trying to use. Or you can do it in an indirect staining, which detects the protein of interest with a primary antibody that then binds a secondary antibody that is conjugated to a fluorophore. So both of these, of course, are possible for you to use. So I mentioned it before, if you have archive slides, you can use these quick reference cards that are, I guess, appearing in the chat as a link. So always take these into consideration if you have placed your own sections or if you want to use archived sections to see, does it fit with the allowable area? So the allowable area is basically what you can align in the cytosol, and that will definitely hit the target that you need to hit on your capture area on the Visium slide. So one new feature of this Visium HD assay is also that you can now use either 25mm wide slides or 26mm wide slides. So if you are used to these and you have them as archived slides, for example, use these quick reference cards in the correct way. So we have one quick reference card for the 25 and one for the 26. So make sure that you use the correct one to see if your tissue will be able to be aligned on the Visium slide. So you can download these from our website, and I think the link has been shared with you. So let's look a little bit more into the workflow steps itself. So we have talked about sample preparation. So basically what you do is you have your FFPE section on your glass slide, you deparaffinize it, stain it, image it, and decrosslink. Of course, if you are performing immunofluorescence staining, the decrosslinking comes at an earlier stage. So after the sample preparation part is complete, we move on to the library construction. So the first step after decross-linking is we hybridize our probes overnight, ligate them once the 16 to 24-hour incubation time is over, and then we go into the machine, into the CytAssist instrument, and release and capture our product that we want, and then we move on with the Visium slide that contains the product to extension and barcoding steps, after which we elute. So we basically come off the slide, go into a pre-amplification reaction and the finishing of the library construction, move on to sequencing, and then, of course, the data analysis. So what happens in these steps is shown here as a graphic. So we have, of course, our probes. They bind to the target site. I've shown you this before, but here it's maybe a little bit easier to see. So we have a left-hand side, right-hand side. They bind the targets. The right-hand side contains the pulley A, where the construct will be captured. And the left-hand side has the 2S read, which is an adapter read. So after this hybridization has been completed, we go into the probe ligation. But of course, in between these steps, we do a washing step to make sure that we do not carry over the probes that have not bound correctly. So we make sure that we only work with our well-bound probes into ligation. So once we move into the ligation, you see this little gap here will be closed. And after this reaction, we can move on to release the probes because we want to capture them, right? So this is what happens on the CytAssist instrument. We put in a digestion reaction, and the target mRNA that we used for our probes to bind is then digested, so we can release the construct that we want up here. And this will be then captured right away where it is by the pulley T-tail. And then after the CytAssist run is finished, we have captured the probe based construct that we want. So basically, what kind of gene are we looking at here? When it comes to the probe extension, this is the next step, and this is critical that you do this right away. So there's no stopping point in between here. Just make sure that you are aware of this, and we move then on into the probe extension. So you see that this is our captured product, and we move on to extend this to have a full molecule that also contains the UMI and the spatial barcode. Then we denature this and kind of break apart this ligation product and elute it from the slide itself. So as you can see here, this is on the slide. And now we move off the slide into the tubes, where we are going to run a pre-amplification to just have enough product to work with in the following steps. We clean up our pre-amplification product and make sure that we look into how much of a product do I actually have. And this is done with a qPCR, and based on that, we decide how many cycles this product needs for the sample index PCR. And after the sample index PCR and the following cleanup, you're basically done. This is what your finished library will look like. It has the adapters. It has two indices. It has the read one, the TrueSeq read one from your oligos. It has the UMI. It has the spatial barcode. It has the capture T-tail, and it has the ligated probe insert. So this is basically after the sample index PCR and cleanup what you're looking at. And of course, what we do recommend you to do is check your library with the bioanalyzer or tape station to make sure that we see this kind of phenotype. So we want the very sharp peak in this area of 240 base pairs, and this is definitely recommended to see if the cleanups have worked fine and if your library has the expected size. When it comes to the sequencing configuration, I told you earlier that the UMI and the spatial barcode together are 43 base pairs. So this has to be put in the length of read 1. And we do not recommend pooling these Visium libraries with other 10x libraries because the read 1 will be 28 there and 43 in this. So the pairing would be, or the pooling would be a little bit tricky. We have 10 base pairs length for the I7, 10 for the I5, and the read insert has the length of 50 base pairs. So this is the recommended sequencing configuration for your Visium HD libraries. When it comes to sequencing depth, if you are familiar with the earlier version of the Visium assay, we had a little bit of a formula how you can calculate how many spots are covered with tissue and how many read pairs per spot you should put in there. Of course, this falls by the wayside now because we do not have oligo covered spots anymore, but we have this continuous lawn that is capturing everything that is put on top of it. So what we say here, the recommended minimum sequencing depth is 275 million read pairs per fully covered capture area. So basically, if you imagine that this whole area is completely covered by tissue, you would count 275 million read pairs as the recommended minimum. The ideal depth will, of course, kind of depend on your tissue type and what kind of experimental question you have. Sequencing deeper can add valuable information a lot of times. So, but naturally, you will not always have the entire capture area covered. So you can use the Loop browser manual alignment wizard to calculate how much of your capture area is actually covered by tissue. So if it tells you, okay, you have 60% of your capture area tissue covered, then you would calculate your minimum base pairs in that way. So you go to 75 million by 60%, and then you know what your recommended minimum read depth would be. So just keep in mind that this sequencing depth recommendation might be a little bit low for your tissue. So make sure that you think about this carefully and maybe look into literature if you need a deeper sequencing there. So I'm going to tease you a little bit with some really nice images. So basically, what are we doing all this work for? So maybe we do it for something like this, for being able to discover crucial molecular differences between healthy and diseased tissues, for example, or to something like this, basically research that can build new foundations of understanding developmental biology, different kinds of biologies that we can go deeper into. Yeah, this is not what I wanted. Okay, but basically to get there, we need to dive into a lot of data. So we are performing spatial transcriptomics in this assay. So at the outset, we already have two types of basics we look at. We look at imaging data and we look at transcript data. So from the images that you take yourself, a high resolution image and an image from that the cytosist takes itself, and from the slide layout, we already have a couple of layers of imaging data that we're working with. On the other hand, we have transcript data. So we have the expression of any gene on your tissue, but we have it with the location encoded because of the barcode that we are attaching to the probes. So we also have the probe sequences, and we have the reference genome that we're working with. So we have a couple of layers of transcript data as well. To analyze all this data, we have different tools that are included in the workflow. So some of you may already be familiar with Space Ranger. So the Space Ranger is basically a collection of pipelines for processing the Visium data. This is developed by us. You can download it without issue, as long as you have a very big RAM available to you. So this could be a server, for example, or this could be a cloud service. So this is a Linux-based command line system. So you will need a lot of coding. Nah, you don't need a lot of coding. You just need to know how to work with these command line pipelines for processing the Visium data. For the Loop browser, this is a software we have developed for the analysis and visualization. So you can use it on your desktop or laptop computers. You don't need the same kind of calculating power as you need for Space Ranger. And of course, we have a lot of community developed tools. So these are not officially supported by us. They are developed by the research community, and they are typically programming libraries with some standalone tools that are directed at a certain type of data and a certain type of question. So there are a lot of these tools, and I'm saying we do not officially support it, which is true, but we do have some recommendations for you on our website. So if you are looking into reaching out the data analysis by yourself, we do have some recommendations for you. So when it comes to the general software workflow, we do use two pipelines every time. So this is the make-fast-q pipeline to convert your BCL files that come out of the sequencer to FASTQ files that you can then use in the subsequent count pipeline. So this make-fast-q and the count pipeline are the two main pipelines that Space Ranger uses to create an output that you can work with. So as I mentioned, the make-fast-q uses the raw output of the sequencer. The count pipeline then kind of incorporates the images that you will put into the analysis and the transcript data, and creates an output for you. And what this output looks like, I will show you in a minute. So just this is a bit of a better representation of what Space Ranger count does, because it is central to the data analysis workflow, because it incorporates both types of data that we worked with, so the imaging data and the transcript data. And this is what that looks like. There is an optional step if you want to look into the alignment of your tissue to the slide itself. So you can go into the Loop browser and do a manual alignment of your tissue to the slide architecture. This is not necessary. This is not mandatory. This is an optional step for you if you think the alignment does not look fantastic. The site assist image, the high resolution image, and the slide layout, all this information goes into this count pipeline together with the transcript data. So the transcript data is the FASTQs that we made, the reference genome, and the set information that we are giving it. All this is incorporated in the Space Ranger count pipeline. And this is why this is so central to your data analysis. The output then can be used to be visualized in the Loop browser, or you can do an additional analysis with community developed tools But this is a crucial output that you need to get on with your data. So we have in the Space Ranger count output, there is a lot of information that you're actually going to get. So we have this structured that you can find it easier. So if you're interested in what that looks, we have everything on our website. We have a web summary file for assessing the data quality. And this web summary file is actually very, very useful just to get an impression of what does my image look like? What does my data look like? Does this make sense before you delve into the deep analysis? The data that are provided is, I mentioned it before, the 8x8 binned results. It also gives you 16 by 16 micron binned results. These are generated by default. But there is a command line option if you want a custom square bin. So if you want your own size, that is not included here. You also will get the 2x2 micron raw matrices provided to you. So if you want to go down to the command line option, you can choose whichever bin size you prefer. We have a spatial folder for all the data that you will get processed, and that where you can take any kind of information and any kind of data type you want and go into community developed tools and look into the image processing, for example, or the secondary analysis. This folder has everything you will need. You will get a loop browser file. By default is the 8x8 and a custom square bin if you have run this, and the secondary analysis information you will also get for the 8x8, 16, and custom square bin. And this is what that output structure that I mentioned look like. So you will have loop files for putting it into the loop browser. You always get the 8x8. You can also get a custom one if you have chosen to do so. You will get an H5 file format for if you want to do your own data binning. You will get a metric summary summary file. You will get the read level information for third-party tools, so you get all the molecule info as well. You will get an overview of the probe set that you have used, the read alignment details and BAM index files. You will also be able to get in the web summary HTML. So this is a lot of words that I've just said. So if you want to take a look at that, I will show you where you can find all the detailed information. Okay, so that was the introduction to the different parts of the Visium HD assay. We have a lot of documentation. So if you are familiar with our website or with our assays, you know that there is a lot of information available to you. I'm pretty sure that during my talk, a lot of links have already been shared with you. But if you go into the support website, you will find a lot of information, for example, for how to prepare your tissue if you are new to this, or if you need guidance there. We have imaging guidelines in a new techno that is specific for Visium HD. So if you are uncertain how you need your imaging set up, this will give you a lot of information and a lot of help. The tissue alignment quick reference cards I mentioned are also just for download on our website. So make sure that you download the correct one if you have 25 or 26 millimeter slides. We have a tissue preparation guide that shows you how you can use archived slides, because I know that archived slides, especially hard-covered slides, can be a little bit tricky, but it will give you some information on how you can prepare these archived slides. But it also, of course, gives you a lot of information of how to prepare your tissue blocks, how to section your blocks, how to place them on the slides correctly, and how to move on from placing to hematoxylin and eosine staining, or to immunofluorescent staining. And then finally, we go into the user guide for the assay workflow and a data analysis page. So we don't have a user guide for data analysis, but we do have a software page. And this is what I want to show you on this slide. So this is when you move to our support website and you move to the Visium HD gene expression. I put a little circle on it here. We have the tab software analysis, and there you will find all information, tutorials, anything you can think of. And what you can do as well is go into the datasets tab and look at the public datasets we have available. Because there you can download everything. You can see the output structure that we have by default from Space Ranger, and you can download the datasets and take a good look at this, what it actually looks like, what you can do with this. So I would recommend to do this if you are very interested in the analysis. This is super helpful. Okay, and with that, I am gonna end my talk.
Thank you so much, Antje. That was very interesting and truly insightful. We have a lot of questions coming up. I collected some for you, but before going into the Q&A, we have a second poll. Here we would like to know if you would like to be contacted by your local 10x Genomics team member for technical information or for pricing, or if you are not interested. So I will launch that one now for you to answer. In the meantime, we can already start with the questions I have collected for you. So there were a lot of participants who were asking if there is also an option to do Visium HD with fresh frozen or fixed frozen samples.
Yeah, so we are working on that. It's coming up. For the time being, the only supported and the only tested tissue type is FFPE, but we're working on it, and we are, as soon as we know how to get to the data from your fixed frozen or from your fresh frozen samples, we are absolutely gonna let you know. And if you know us, you know we're gonna push out a lot of protocols. So I hope these will be coming soon.
Perfect, thank you. So then we had a question from Damien. He actually asked you if Visium HD can be also performed on already sectioned and stored FFPE slides, and you answered it directly on the next slide. But then he also asks if there is specific equipment needed to calculate the DV200.
Yeah, so all the equipment that you need and all the kits. So there's a QIAGEN kit, for example, where you can go into and get the RNA out of very low amount of FFPE tissue. If you need the exact catalog number, I cannot give it to you, but we do have it in our tissue preparation guide and in our guides for how to do the DV200 for archived slides and for blocks. So wait, can I go back? No. This is one of the, no, I cannot. Okay, sorry, I wanted to go back to my slide. So this is in one of the overview protocols that I shared.
Then there came another question from Damien. He asked about stopping points, specifically after the decross-linking, if there is any storage option. For example, if he wants to prep the slides in his lab and then take over to a collaborator with a site assist elsewhere.
So not after decross-linking, because then you make your tissue a little bit unstable. So after decross-linking, you have to move on directly to the hybridization part. But there is a stopping point when you want to go after imaging, for example. So this is basically after imaging, before decross-linking. Then there is a safe stopping point for you to, I don't know, walk across a campus or however far away it is. So that is the safer stopping point. And you have to keep in mind that the decross-linking, for example, is already done on a thermocycler. So if you want to perform all the thermocycler in steps in one lab, then I would move it after the imaging.
Perfect. Then we had a question from Sarah. She asked if it's possible to include the full resolution microscope image into the loop browser for Visium HD.
So you mean the high resolution one? that you take in the beginning. Oh yeah, absolutely. You can use it put it into the count pipeline. So you can use your high resolution image, you can use a site assist image, and of course you need the slide layout as well. So this is the image files we're working with.
So because we have some minutes left, I will also pick a few more from the list. Albert, for example, asks if there is any plan to have different Visium HD panels of other species than human or mouse.
I don't know how much of a plan it is. I know it's a feature that a lot of people are interested in. So I don't think at the moment we are planning anything super precise in that. But if you want this as a feature, we are always happy about feedback from you.
Perfect. And then we have another question. I just need to check which one to take. There was a question if when we are upgrading the firmware of the site assist, if it's still possible to run non-HD samples on it.
Oh yeah, absolutely. It's just an upgrade to also incorporate the HD. You will be able to run the old essays as well. That's not a problem.
Hey, I think there are no more questions actually. I think let me check. Here we have another one from Luisa. Is it possible to do IHC instead of H&E or IF before running the Visium protocol?
That depends on the protocol. So in these cases, we kind of need to take a look at your protocol and see if this would be compatible. So, but this is what our amazing support team is for. So if you have very specific questions and you wanna discuss the details about this, we are very happy to help.
Perfect. And another one coming for you from Zimming. He or she, actually, I don't know exactly for the name, was a bit confused why we cannot stick to the histological slide with a barcoded slide by hand and why it is necessary to use the site assist for Visium HD.
So you mean direct placement onto the slide? Yeah, that would be very tricky. So the slide needs to be prepared in a certain way. So this would have been too much detail for today's talk. But the thing is, I showed one. If you end the poll, I can actually go.
I ended it. Maybe you can go back.
Okay, maybe I can go back a lot. Let me go. Sorry. Oh, yeah. There's a lot there. So the point here is that the site assist makes it possible to kind of direct the capture of your transcripts that you want to have. If you place your section just on the slide and you have nothing here that kind of focuses the target capture to a certain area, you have loss, and targets can be released somewhere else where you don't want them to be. If with the site assist you direct this onto the exact area where you want it, and it's in a small kind of cleft between these two slides. So this is why this is important. If you have a high resolution like this, you don't want anything to swim around uncontrolled. So this is why the site assist is necessary to have this absolute controlled capture for Visium HD.
And then Damien asked again, are 8 to 10 micrometer sections compatible with Visium HD, or what is the recommended section thickness?
So at the moment, the recommendation is 5. Still 8 to 10, I'm not sure. So I know we have tested different thicknesses, but the exact range for HD, if it goes up to 10, I would have to check. But I can get back to you about this.
Okay. Let me see if we can find another question. Is it still possible to use Dacro-Meyer's hematoxylin for the H&E stain?
Yes. So it's still possible. It still works. But we've seen that the GIL just gives us much better morphology images.
And with the last question, there came another question. Can we get desired results from 14 micro thickness sections?
Ooh, that's tricky. I wouldn't recommend to use those. Kind of depends on what tissue it is, but 14 seems way too thick because then we don't really have control over how much the permeabilization and the RNA digestion works and how deep they go into the tissue in hybridization. So this is very very unsupported, and I'm not sure we have tested this even. So this is definitely not recommended.
Okay, about the thickness, there was coming an answer from Bart from our panelist, 3 to 10 micrometer thickness.
3 to 10, okay. I wasn't sure it was 3 to 8, but 3 to 10, that's very good to know. Thank you.
Okay, perfect. So I think all the other questions were answered already by the panelists. So we are 5 minutes earlier today. If there are more questions coming up, feel free to put them in now. We have 5 more minutes. Otherwise, we will leave you out a bit earlier for lunchtime. We are very happy that you joined. And the person who won the gift card and answered all the questions in the poll will be contacted by us via email. So stay tuned for that. Don't miss the email. Thank you again, Antje, for the presentation, and I wish you all a fantastic day.
Thank you so much, guys.
Bye.